So this is the second part of the previous video, which was the video on the preparatory stage of the glycolysis. In this video we are going to have a look on the energy yielding reactions of the glycolysis. In the previous video uh, looked the we looked the splitting of the fructose 1,6 bisphosphate by aldolase enzyme. Two molecules were produced, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This dihydroxyacetone phosphate was further converted or isomerized into glyceraldehyde 3-phosphate by the triose phosphate isomerase enzyme. So at this stage we have two molecules of glyceraldehyde 3-phosphate, and both of these molecules will continue down the pathway. So the next reaction is the conversion of this glyceraldehyde 3-phosphate into the 1,3-bisphosphoglycerate. So the glyceraldehyde 3-phosphate has a phosphate group attached to its third carbon. Now another phosphate is going to be attached to this molecule at the first carbon. But notice that this phosphate is not coming from ATP, this phosphate is coming from the inorganic phosphate pool of the cell. Moreover, the cell is also using another molecule in this reaction, which is the NAD positive. This NAD is going to be reduced and it is going to be converted into NADH, because this is an oxidation reaction. The conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate is an oxidation reaction, and in this reaction the hydrogen present at the first carbon of this molecule is going to be removed and it is going to be added to the NAD positive, and that hydrogen is being replaced with the inorganic phosphate. So this makes this molecule 1,3-bisphosphoglycerate. So this hydrogen is removed, this bond is cleaved, and this phosphate is added here, and this hydrogen is added to the NAD positive. So the reduction of NAD takes place. So in the reactions in which NADH or NAD is taking place, they are catalyzed by dehydrogenase enzyme. So this reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, and the product of this reaction is 1,3-bisphosphoglycerate. So here I want to mention an important point about the nomenclature. Whenever you see the prefix bis with any other with the name of any other molecule, it is indicating that a functional group is added to both the terminals of this molecule. So in this case a phosphate is added, so the phosphate is added to the first and the third carbon of this molecule. So two phosphates are added, and this is a bis molecule, which is indicating that these phosphate groups are added at the terminal of these molecules. If this molecule were 1,3-biphosphoglycerate or diphosphoglycerate, then it would be that two phosphate groups are present on this molecule, but their location is not at the terminals. And other thing is that NADH is going to be used in the electron transport chain, where it is going to be oxidized, and again the NAD positive is going to be replenished, and the cell can use this NAD positive in this glycolysis reaction. So the next reaction is the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate. So in this reaction ATP is going to be produced. Cell is going to transfer this phosphoryl group at the first carbon, and and it is going to be added to the ADP, and as a result we get an ATP molecule and 3-phosphoglycerate. The phosphate from the first carbon is removed, and there we get a carboxylate ion. This reaction is catalyzed by phosphoglycerate kinase, and kinases are enzymes that are involved in the transfer of phosphoryl groups from a substrate molecule to ATP or from an ATP to a substrate molecule. Next reaction is the isomerization reaction, in which 3-phosphoglycerate is isomerized into 2-phosphoglycerate, and this reaction is catalyzed by phosphoglycerate mutase, and mutases are enzymes that uh, that are involved in the transfer of a phosphate group within a molecule. Kinases are involved in the transfer of a molecule from one molecule to the other molecule, but the mutases are involved in the transfer from one location in a molecule to the other location within that molecule. The significance of this reaction will be illustrated in the next reaction. In this reaction, 3-phosphoglycerate is converted into phosphoenolpyruvate. So in this reaction, the low energy phosphate ester bond of 3-phosphoglycerate is converted into the phosphoenolpyruvate, or high energy phosphate bond of the phosphoenolpyruvate. In this reaction is catalyzed by Enolase enzyme. So the 3-phosphoglycerate is converted into phosphoenolpyruvate, and water molecule is removed, and these water molecules come from the hydroxyl group of the third carbon and the hydrogen from the second carbon. They make up a water molecule, and we can see that there is a carbon carbon double bond between the second and the third carbon. This is the enol functional group, or enol uh, component of this molecule. So enol is a functional group in which a double in which a double bonded in which the carbon carbon double bond is linked to a hydroxyl group. In the previous reaction, you can see that these two carbon molecules, there is a here you can see that this carbon is attached to the hydroxyl group, and in the next reaction, the there is a and in the next reaction, these there is a double bond between the carbon 2 and carbon 3, and the hydrogen of the hydroxyl is removed, and a phosphate group is replaced a phosphate group is added at that site. So this is the high energy phosphate bond, and why it is high energy we will see in the next reaction, which is the last reaction of glycolysis. In this reaction, phosphoenolpyruvate is converted into pyruvate, and this reaction is catalyzed by pyruvate kinase, which transfers this phosphate from the second carbon to the ADP, and it forms ATP. The remaining molecule, the molecule that is left after this reaction, is the enolpyruvate. Enolpyruvate is a tautomerization form of pyruvate. So here we can see enol and enol ketone tautomerization. The enol form of pyruvate is converted into a ketone form of pyruvate, and the keto form of the pyruvate. So this is the enol form of pyruvate, and it is the keto form of the pyruvate. When this phosphate is removed, the enol pyruvate undergoes stabilization and resonance stabilization. When this phosphate is removed, this enol pyruvate is undergoing when this phosphate group is cleaved, the enol pyruvate undergoes resonance stabilization, and it releases a lot of energy. That energy is also making this bond this compound a high energy compound. So that energy is also going to be used to make ATP from ADP, and this phosphate is added to this ADP, and ATP is produced. And also notice that this reaction is also irreversible. So after the splitting reaction, we had two glyceraldehyde 3 phosphate molecules. Each of the glyceraldehyde 3 molecule will follow or further along the pathway, and each glyceraldehyde 3 phosphate molecule produces 2 ATPs. So when two molecules of glyceraldehyde 3 phosphate will end this pathway, we will have 4 ATP molecules. But also remember that 2 ATP molecules were utilized in the preparatory stage of this pathway. So the net ATP that are produced after the glycolysis are 2 ATP molecules, and those 2 ATP molecules are going to be used as an energy molecules. In the next video, we will look at the substrate level phosphorylation, and also the what are the fates of pyruvate and the energy yield of the glycolysis. Thanks for watching. Allah Hafiz.